All right, I think we'll begin. Thank you all for coming. We are very excited that you're here with us today. And we really just want to share hope and share what we're doing and let you know how research is going on and also introduce us if some of you don't know us. So, Danielle Meadows, who I will introduce in just a minute, lives near Boston, Massachusetts, and I live near Los Angeles, California. And we're very excited that we're a global all over the world, and we know a lot of you in Europe, and I probably have met some of you and had the pleasure of meeting some of you on some of my trips to Europe over the years. And you may know that I'm a parent and share your urgency, and we really want to share the research and tell you how much it's moving forward and that we're continuing to push ahead as fast as possible. And we're doing these webinars really a few of them through the years. So stay tuned. We'll give more during the year to let you know what's going on. So for those of you that may not know us, I'm Linda Tannenbaum. I'm the CEO of Open Medicine Foundation, OMF. And it's a global research-focused nonprofit dedicated to ME/CFS and now long COVID and the related multi-system chronic complex diseases that are very similar and have very similar symptoms. And over the past 13 years, we've established 7 collaborative centers, as you see, all over the world, and including our center here at Uppsala in Sweden with director Dr. Jonas Bergquist, who collaborates with all the others in the UK and Netherlands and Germany and Finland and others. And then also at our collaborative centers, as you see at Stanford and Harvard and University of Montreal in Canada and University of Melbourne in Australia. And we also partner with Bateman Horne Center for Medical Education in Utah. And we're tackling these diseases from many angles for the purpose of finding effective treatments and a way to speed up the diagnosis and ultimately a cure and, of course, prevention. So our work is patient-centered and data-driven and guided by the principle that open collaborative research is the most important way to get answers quickly. So open collaboration, open global collaboration. So we also offer helpful resources that you can use that are already on our website, educational materials for clinicians and families, and so take a look at it if you want to bring something to your physician or show your family or get some insight into some suggestions that are from others, from treatments and diagnosis. And also take a look, there's updates on our funded research studies and the opportunity to be part of the solution by signing up to our OMF Study Me registry, which Danielle will talk more about. So I just want to go over just real quickly what our purpose and mission and vision is. We revolutionize the way that we understand and treat chronic complex multisystem diseases with the initial focus on ME/CFS and long COVID by illuminating their mechanisms, accelerating effective treatments, and empowering personalized care. And our mission to drive and fund this global collaborative research to unlock disease biology, deliver precise diagnostics and advanced treatments, giving every patient a pathway to care and hope. And our vision is a world where ME/CFS and long COVID and these related diseases are understood, rapidly diagnosed, and effectively treated, so every patient can live fully. So this is our picture and our commitment. As we said, we have 6 collaborative research centers at 5 universities in 4 countries in 3 continents. And we have our Medical Education Resource Center with Bateman Horne Center, which we call MERC. We have 21-member scientific advisory board helping really decide what research is important and giving their insight and expertise in everything that we're doing. We have OMF that we established in 2012. We have a entity of OMF Canada in 2018 and OMF Australia in 2021. And we have 3 foundation boards and which really oversee all of our governance and oversight. So I want to thank you very much. And I'm going to introduce Danielle Meadows, our Vice President of Research and Operations. And she'll share some insights into our research portfolio. So Danielle holds a PhD in biomedical engineering and has dedicated her career to improving people's lives through advancements in clinical research. And I'm going to stop sharing and hand it over to Danielle.
All right, thank you so much for the introduction, Linda. I will share some of my slides here. Okay, so as Linda mentioned, I'm going to spend a little bit of time talking about OMF's research portfolio, including a high-level overview of the portfolio itself, some examples of OMF-supported projects within that portfolio, and then a somewhat tangential piece that Linda mentioned already, being the Study Me registry. So starting out with an overview of how we're trying to have this holistic research portfolio. So at its core, there are three pillars of our research portfolio, which are somewhat reflected in the purpose and mission and vision that Linda just read through for you. So through our research, we aim to identify clear disease mechanisms, enable accurate diagnosis, and find effective treatments. And right now we have around 40 active projects spread across our 6 centers, and they include a large number of collaborations across those centers, like Linda mentioned, really focusing on that global collaborative, not as siloed way of going about research. So I want to give you a quick flavor of some of the work we have going on at our centers and across these three pillars of our portfolio. But of course, with 40 active projects, I can't talk about every single one of them. So I'm just going to give a couple of examples today. But if you want to read more about these projects or any of the others we have going on, I do try to keep our website as up to date as I can with information that we're able to share publicly. So feel free to, peruse at will. So again, we have these three pillars. And before I talk about global OMF, I just want to give a quick highlight of some of the work going on at our collaborative center at Uppsala. So this research is directed by Dr. Jonas Bergquist, who has a lot of expertise in neuroinflammation, immunology, proteomics, metabolomics. And so, with this focus on the disease mechanism, his team is doing a lot of in-depth analysis of neuroinflammation, studying cerebrospinal fluid, trying to understand how herpes simplex encephalopathy might lead to ME/CFS down the line, evaluating the development of ME/CFS from COVID, studying more severe patients from their homes, and then gaining a deeper understanding of the kynurenine pathology as well. And in recent years, we've been, shifting a lot more work into the diagnostic and treatment aspect of things. And so in this realm of diagnostics, Dr. Bergquist is looking at some specific markers related to the central nervous system. And then he's also a principal investigator on our BioQuest project, which I'll talk a little bit about later on. And then in the treatment area, he's also really heavily involved in our life improvement trial, or LIFT, which I'll also talk about a little later on. So we'll get a little bit more in depth on those two projects. So going back to global OMF, I mentioned wanting to give you a snapshot of work going on across our centers and then within these pillars. So I'll start with identifying clear disease mechanisms. And we have a few approaches to this that are kind of ranging from looking at overarching mechanisms of ME/CFS as a whole to then mechanisms of some of those hallmark symptoms. So on the latter end of that spectrum, I want to start with a study that's investigating one of the more under-researched symptoms of ME/CFS, so unrefreshing or non-restorative sleep. And this study is being conducted at Beth Israel in Boston. It's in collaboration with our Harvard and Computation Centers, and it's a prospective, in-depth sleep study on people with ME/CFS and long COVID. So it's studying a lot of different aspects of sleep, including sleep circadian dysfunction, immune function, brain electrical activity, and mechanisms of state boundary control. And that's just the transition between sleep stages, so going, from REM sleep to non-REM sleep, and then neurodegenerative diseases as well. And I think an important aspect here is that these data are also going to be combined with a retrospective look at polysomnography data. And these recordings have been used to show some preliminary results that we're hoping to confirm with this prospective study. And so in brief, I'll just say that the preliminary examination of these data show increased fragmentation and sleep spindle deficiencies in people with ME/CFS. So then stepping back to look at the disease as a whole, I want to briefly talk about some work from our computation team that's called Network Medicine. So network medicine looks at the interactions of genes, proteins, pathways, and other biological components. And so it's this network of things, hence the name. And it uses those interactions to better understand a disease. So in this context, our computation team is combining published preclinical and clinical data from ME/CFS and similar diseases to help identify potential candidates for research. And I think one great thing about this approach is that the team can actually periodically retrain the model as new and updated information comes out. So the hypotheses that it's generating really actually evolve and help stay up to date as new things come out. So then going back to our pillars, I want to move on to the accurate diagnosis portion of the portfolio. And it's probably obvious from the name, but the ultimate goal of our research in this area is to find an objective way to diagnose ME/CFS, including differentiating it from similar diseases. So I'll tell you about two projects that we have going on in this area. And the first is actually from our collaborative center at Stanford University, and it's developing a neutrophil assessment platform. So this is a platform that facilitates isolation of neutrophils from whole blood and then artificially simulates an inflammation event. So then before and after this event, the team compares ME/CFS and healthy patients' neutrophils, and they look at things like apoptosis, so cell death, migration, reactive oxygen species, and neutrophil extracellular traps. And the idea here is that neutrophil dysfunction is an indicator of chronic inflammation. So you would expect that neutrophils from people with ME/CFS would behave differently from those of healthy people because we're expecting this chronic inflammation to be happening. So given more development and testing, it's possible that this technology could eventually be used to diagnose ME/CFS in the future. And then the second project I want to mention in this area is our large-scale biomarker project that we call BioQuest. And the goal of this study is to identify a biochemical signature or biomarker for ME/CFS that can be conveniently evaluated through a blood test. And importantly, it would differentiate ME/CFS from other illnesses with similar clinical presentations. So we are launching phase 1 of this project very imminently, and it'll include multi-omics, cytokine testing, immunoassays, all on 1,000 plasma samples to try to identify this biomarker signature. And in this project, we are able to incorporate disease controls like multiple sclerosis, clinical depression, and burnout syndrome. And with our sample size, we're really hoping to do some in-depth subtyping analysis since we know that not all ME/CFS looks the same in everybody. So then now we're on to the final pillar here, which is finding effective treatments. And I think an important part of our approach to this pillar is that, again, we recognize the heterogeneity of ME/CFS. It doesn't look the same in everybody. And we are hoping to approach finding treatments from multiple angles that kind of address that concept. So I'm going to spend a little extra time talking about personalized medicine before I wrap things up today. Personalized medicine is a way of approaching treating patients that's based on specific indicators of what might work for that individual person. So I'll describe maybe two ways that we're approaching this. So we can look at it from the molecular level, so what's going on from the molecule level, and actually how that translates to the clinic. And then you can also look at it in the reverse direction. So what do we see in the clinic, and then how do we figure out what's going on at the molecular level to match that? So thinking about first going from the molecular level to the clinic, we have a project at our Montreal Center, which is focused on hypothesis generation for treatment trialing. So they're approaching this through 3 steps, which are to really go through some deep phenotyping of ME/CFS patients with an emphasis on this precision or personalized medicine, exploring past clinical trial samples to gain some new insights where possible, and then doing some in vitro validation or petri dish kind of studies of FDA-approved drugs for repurposing them as targeted therapeutics. So If you maybe keep tabs on publications from on ME/CFS or long COVID, you may have seen some of this work recently that was published, and it shows that a molecule SMPDL3B is elevated in the plasma of some people with ME/CFS. So an example here is that the team is now exploring new therapeutic approaches to address that elevated level of that molecule in the plasma. So now thinking about the other direction, going from the clinic and trying to figure out what's going on at the molecular level. At our Australia Center, for example, they're conducting a personalized treatment trial, which is aiming to learn from a general practitioner as they trial different treatments on patients. So this study is really trying to deeply characterize patients before, during, and after treatments that they try. And they'll try to identify patterns in each person's biology that corresponds to the changes and symptoms that are induced by a specific treatment. And so ultimately, the goal is to identify clusters of patients with specific biology symptom dynamics to give a better approach to treatments in the future. And the final project I'll mention today is another one going from the clinic to the molecular level as we work towards this personalized medicine approach. And that is our life improvement trial, or LIFT, which is our clinical trial evaluating low-dose naltrexone, or LDN, and pyridostigmine. And the diagram that I show here gives just a graphical representation of the protocol. But what I want to emphasize are actually the blood samples taken before and after treatment. And a really important part of this trial involves those particular blood samples. We know from people's experiences that LDN and pyridostigmine won't work for everybody. So we want to be able to use those blood samples to do some extensive testing that will hopefully allow us to learn what makes somebody a responder or a non-responder to the treatments. So again, kind of moving on this idea that the heterogeneity of people with ME/CFS indicates that personalized treatments are potentially going to be a more effective way to move the needle. So I've thrown a lot of things at you so far. So to recap quickly, OMF's research portfolio consists of these three major pillars. So within the pillar, trying to understand the disease mechanism, I talked a bit about our sleep study, which is looking at the mechanism of a specific symptom, and then network medicine, which is looking at ME/CFS as a whole. In the diagnosis pillar, I talked about the neutrophil assessment platform as a potential new diagnostic technology, and our large-scale ME/CFS biomarker project called BioQuest. And then finally, when thinking about effective treatments, I focused on our approaches to personalized medicine with a hypothesis-driven approach, taking molecular changes and trying to identify treatments to match them. And then two ways of taking clinical responses to treatments and trying to identify the corresponding biology that could be predictive of those responses in the future. And here, I just want to extend kind of beyond individual projects and give a quick visual representation that tries to show kind of how research on ME/CFS connects to each other. So from this really high level, in the top left of this diagram, it shows that there's some combination of triggers and risk factors that a person could fail to recover from. And that would place them into this web of system dysregulations and other factors that kind of all together yield this disease phenotype. And how the different components of this web interact can yield varying symptoms, which can help explain how different people have different clinical presentations of ME/CFS. All right, before I turn things back over to Linda, I want to spend just a quick minute switching gears to talk about this other component of our research endeavors, which is helping to bridge the gap between researchers and patients. So if you haven't seen this yet, OMF has a patient registry called Study Me, and it's a collection of people around the world who are interested in participating in research on ME/CFS, on COVID, fibromyalgia, and other post-infection syndromes and things of that nature. So on the slide, you can see how registration in Study Me has grown a lot since its launch in April 2023. We now have over 14,500 participants around the world. And we collect some basic information from people during registration, like their location, age, sex, and types of studies that they'd be interested in joining. And it's really all with the goal of being able to send people studies for which they might be a good match. We don't want to spam your emails all the time, but really trying to make sure, bridge the gap between the researchers and people that might actually be able to participate in that research. So we've tried to make it a low effort to register while still being able to deliver more relevant opportunities. And we're now at a good spot where we're reaching out to researchers a little bit more actively to help recruit for more studies, really trying to accelerate research however we can. And so far, we've helped recruit for institutions like the University of Melbourne, Stanford, Harvard, Scripps Health, Bateman Horne Center, and more. And with that, I will say thank you so much for listening. Thank you for being here, and I will turn it back over to Linda.
Thank you, Danielle. That was excellent. And I know that there'll may be a lot of questions that if you'd like to enter any questions in the Q&A, I'll go through those and we'll answer them the best that we can. And if you have questions afterwards, you're welcome to email us at any time. And, you know, as Danielle said, we have so many projects going on. We just wanted to highlight some of the more exciting ones that you might want to know about, but there's a lot of research going on behind the scenes, and we're really excited about it. We've had more publications this year than we have in any other years because, as you know, research takes a long time, and so a lot of them started years ago, and this year they came to fruition, and there's a lot of publications. And the benefit of publications, of course, is a lot of people learn from what we're doing, and then it opens more collaborations with other people all around the world, and the research is shared in a very big way. And then other researchers start collaborating with each other. So it's really ignited research over the years. I mean, I started this 13 years ago, and the research in the last 3-4 years has really grown exponentially. And so there is a lot happening, and we know that you're all waiting for answers and treatments and a way to diagnose this and validate this, and it's getting close. And so we are very excited about all the research that's happening, and we hope to have some more information and breakthroughs in the next year and a half. So I'm going to go through the Q&A, and you're welcome to continue to ask any questions that you have. First one, which is lovely, says season's greetings to all, and yes, happy holidays and season's greetings to all of you. And thank you for some of your lovely comments in the Q&A. We did have a very big successful year with our fundraising, and we thank you all for being a part of that, because that's the way this research keeps accelerating. Are there any repurposed medications that are being trialed right now? It's one of the questions. Danielle?
Yeah, so the LIFT clinical trial that I was just talking about is looking at two repurposed medications. That was the low-dose naltrexone and pyridostigmine. So both of those are FDA approved, and we're testing them as repurposed drugs. And, you know, things like the personalized treatment trial that I also mentioned are using, you know, repurposed drugs as well. Maybe one thing that I will share quickly. So we this year had published a paper that's on patient-reported outcomes of different treatments. So I can actually just, I'll share my screen again really quickly. So if you haven't seen this, this is a paper that was examining patient-reported experiences with over 150 different treatments. And I think they had about 4,000 respondents to the survey. And so this was, you know, some analysis that our computation team did. The survey was conducted by a patient researcher. And so what this figure is kind of showing here that you can see is these are the top 20 treatments that as described by patient experience. And over here are the symptoms that they felt like each treatment really had an impact on. So for example, looking at, you know, the top one, fluids and electrolytes, in general, orange means better response, blue means bad response. So they had a pretty net positive reaction to using fluids and electrolytes, and people felt like it helped most with postural orthostatic tachycardia, as you might expect. But there was also some effect on fatigue, feeling worse after, so post-exertional malaise, brain fog. And so if you want to explore this paper a little bit more, like I said, there are over 150 treatments that patients provided information on and what they felt like had an impact on their symptoms. So this can be a hopefully helpful resource for you all.
Is there a way for us to put that in the, so that they
Yes,
can
I am
click the
going
link?
to do
Okay,
that now.
okay, great. Okay. All right, thank you. Sensitive patient data is difficult to manage across a decentralized network of researchers. What are some of the local and global compliance regulations to be met when collaborating on private and sensitive data sets?
Yeah, that's a great question. It is challenging. So some countries, in particular, like data collected in that country to be housed on that country's servers, for example. That is, it's a kind of country by country nuance that you have to take into consideration when collaborating across different centers. And in many cases, it's a matter of setting up the appropriate paperwork to make sure that any data sharing that occurs is both secure and consented for by the patients or the people that participated in whatever study is going on. So it is quite complex to do, but I think at this point amongst our centers, we at least have things pretty oiled out. So we're sharing data safely and securely, but rather efficiently at this point.
Thank you. Will these slides be sent? Yes, we're going to be sending the recording, which includes the slides, to everybody who signed up for the webinar. What role do wearables play in your research? How do users share data with research teams?
Great question. Wearables are one of my favorite topics. I think there is so much untapped potential in them. So I'm very excited that we do have wearable data coming in with our clinical trial. So those are going to be some secondary outcome measures for the trial where we're looking at things like, step count, heart rate variability, all these sorts of really continuous metrics that you can get across the course of a study as opposed to discrete time points. And so I think that that's going to be some really rich data. And we have a number of studies that collect wearable data as well. It's usually through some kind of digital trial system where they can have their wearable and connect it straight to a platform, so they don't have to think about sharing the data. It just kind of happens in the background automatically after they've set up the connection. And so it's usually pretty simple for participants to share their wearable data.
How many subgroups may there be?
That's another great question. So I think it kind of will depend on the data. We have a couple different ways to approach something like I think this is probably in relation to the BioQuest study. So the patient-reported treatment outcomes, for example, that I just mentioned, they did some subtyping analysis with that work and found generally four subgroups that kind of show a different symptom profile. So, one approach to looking at BioQuest would be to take a similar kind of clinical picture and look at those sorts of subtypes and see if there is a biomarker signature associated with those. But then there's also the data-driven approach where you look fresh at the data and kind of see what subgroups come out of it. And in that case, we have no idea how many subgroups there are going to be. So, it depends on which approach you take. We're planning to take both approaches and really kind of see what falls out.
It's very exciting because with artificial intelligence, there's so much more that we can do now. And so before, of course, before they started using artificial intelligence, everything was done in the old-fashioned way, and you could look at just so much large data sets. Now with artificial intelligence and all of the different software packages that are coming out using artificial intelligence, we will really be able to subgroup in so many different ways that we never would have thought of before, combining the clinical data and the laboratory data now. So we're very, very excited about it. It's just a perfect time to start looking at all of this large data, which is why we're really excited about BioQuest, which will have 1,000 samples. And it's a very large study, probably unlike any other study ever done at this level. So we're really excited about what we will learn about subsets. So thank you. Very good question. just to read one of the Q&As, it says you're bringing hope to the dark, and thank you very much. That's our logo, and that's what we're always hoping to do to everybody, because we're really hoping all of us can find answers soon. So thank you. Do you study small fiber neuropathy and ME? It is a non-length dependent. It is non-length dependent in ME. Thank you.
Yeah, so our Harvard director, Dr. David Systrom, has some specific expertise in this area. So in many of the studies that he works on, they do include some analysis of small fiber neuropathy. So yeah, it is incorporated in our studies. It's not necessarily a specific focus of any one particular research project, but it is definitely incorporated.
Yeah, thank you. What is your most groundbreaking research to date?
Ooh, what a loaded question. Gosh, I mean, I think there's so much you could choose from. We have such a wide range of research, it's hard to answer. I think, we have some potentially really groundbreaking work in the diagnostic area coming up. A lot of that still needs to be developed further, but I think it's really exciting. I think this clinical trial is or if not the largest clinical trial in this population to date. And so that is obviously very exciting, but ongoing. And then I think in the mechanism area, we have so many different kinds of studies, from genetics at our Stanford study looking at, this big data study, to, you know, some of the more recent genetics work coming out of our Melbourne collaboration. And then we have this sleep study that I think is incredibly important because it's just so under-researched. So, I mean, I'm saying so many things because it's hard to choose just one, but what I find really important is how they all connect to each other, and that it's not just these siloed results. We're trying to really bring them all together to have a better holistic picture and really improve patient outcomes. That's obviously at the heart of everything we do. So, yeah.
And just adding to that, thank you. There are so many different projects, but we meet with our directors of each of the collaborative centers every month, and then we meet in person once a year. But most important is they talk about what they're doing, and they, as Danielle said, they're really all related. And so the conversations are they're learning from each other, as well as working with each other to figure out what's going on. So the collaborations are the most important thing here, because each person has their expertise, and talking, somebody has expertise in sleep, somebody has expertise in metabolomics, somebody else has expertise in proteomics, but talking together and collaborating together is the most important aspect of what we are all about here. So there's so many different projects that are happening in all different realms. So anyway, thank you, Danielle. What are the plans for next year?
So many things. I think I talked about our three pillars, and I think I mentioned briefly really trying to move not just in the mechanism area, but really start to focus a little bit more on those diagnostics and the treatment areas. We really want to focus on hypothesis-driven treatment work to make sure that we are trying to get as much valuable scientific data for you all to hopefully be able to, bring to your doctors, or bring to the doctors so that they can know it ahead of time, and hopefully help to get some of these more personalized treatment strategies be as scientifically backed as we possibly can.
Thank you. If you have this at your fingertips, what are the doses of LDN and pyridostigmine that we're doing?
Yeah, the LDN dose is 4.5 milligrams per day, and we're titrating up to that over the course of the study. And we are also titrating up to the final dose of pyridostigmine, which is 60 milligrams 3 times a day, so 180 milligrams total per day.
Thank you. I'm in France, and my specialist refused to give me LDN. How could we have access to this medicine? This is a difficult one, but this is really the reason why we're doing these clinical trials, so that we could have a published paper to show how many people it really benefited, so that you could bring it to your doctor and give them the reason to give you the LDN. So unfortunately, we don't have a way to offer or provide LDN outside of France, in France, or outside anywhere. We don't have that access ourselves. But really, that is the reason that we're doing these clinical trials, so that way you could bring this to your doctor and show them why you should be giving those drugs. So hopefully in the future, you'll be able to get this. They're also doing some LDN trials in long COVID. So there'll be hopefully information and publication from both long COVID and ME/CFS separately. Do you have an idea when BioQuest could be ready to be used, at least in the clinical and science realm? Thinking about the beginning of the German decade of research.
Yeah, so I mentioned, we're hoping to launch our phase 1 very imminently. We're just in the final steps of getting all the paperwork finished to get samples shipped to the appropriate places for testing. And from a broad strokes, I would expect testing to be done, mid 2026, and then of course there's a really extensive analysis period after that, and we would have to do verification work and validation work on whatever signature we find. So I know it's not necessarily the answer we all want to hear, but it is going to take a little bit of time before we have something that's clinically ready. That's the unfortunate work of having to start from a discovery stage and go through all of the validation work. But I think, thinking about that beginning of the German decade of research, potentially that's something to explore for some of that validation work. That is
Yes.
definitely going to be a really important body of work. And I think something like that German decade of research could be very valuable to participate in something like that.
The benefit of BioQuest is the samples are already collected. And so, unlike other studies where you have to take 2 or 3 years to recruit patients and then gather samples, this is really low-hanging fruit that we're doing here because these samples are already collected. So the testing itself is exciting because it will be in the next 6 to 8 months. We will have results of the tests, but the analysis of that and then the validation of that, of course, will take time. But we will know a lot in 2026, and we're very excited about this. Just a second, I'm just taking a look here. I don't know if in the chat they saw the paper. You put it in the Q&A maybe?
I put it in the chat to everyone. Yeah. So
And-
the link to the publication that I showed the figure from should be in your chat.
And can they have access to chat?
Yes, they should be able to see it.
Okay, because somebody asked that they said they didn't see the paper, just checking. My main symptoms are pain, specifically neuropathic pain. It is a cluster of ME, and what type of treatment corresponds to this kind of ME?
Mm.
Those are tough for us to answer since we're not in the clinical realm to be able to answer something like that. But you may want to take a look at the Treat Me publication and see if there's anything in there specifically for that. That's really all that we could say at this point because we can't give clinical, we don't even have access to the clinical information right now to help with that, unless you have anything to add, Danielle.
Yeah, we can't give personal medical advice, but I will say if you're able to bring something like that paper to your doctor and work with them, I think there's some useful information there. I mentioned, you can see what some of the other people have experienced going through certain treatments and what kinds of symptoms that they helped. So potentially looking through that and trying to explore how it matches your own symptoms with your doctor's guidance, I think could potentially be a good route to go.
These are great questions. Thank you for continuing to add questions here. I'm not familiar with this, but maybe Danielle is. Have you considered using zero knowledge identity for your patient data?
I am only familiar with it in passing. I think in general we don't take that approach because there is such an extensive informed consent process for each of our studies that we really want to get that really thorough clinical picture and have demographics data, especially for some subtyping analysis and things of that nature. So typically we don't necessarily go through that type of process. That might be beneficial for like a registry, for example, at least in my cursory understanding of what it is. But for our projects in particular, we do try to get as much data as we can through that informed consent process. And then of course, de-identify it for analysis purposes and things like that. But yeah.
Okay. Greetings from Helsinki, Finland. Hello, Helsinki, Finland. What are the biggest roadblocks to advance in research and what gives you the most hope? What are the most important ways that supporters of OMF can help? Thanks for all of your important work. Thank you.
Oh, that's a great question. Wow. Roadblocks to advances in research. I think I'll speak from a general perspective. There is so much to explore in the realm of ME/CFS, with it being such a complex disease and having so many systems involved in the body that I think, crossing across expertise, people always come with their particular background and look at it from a different perspective. And so trying to bring all of those together into one perspective is a challenge, but it's also, I think, can also kind of show some hope in that, we do have these people with all this different expertise coming together to try to solve this problem. And there's general roadblocks to research always, making sure that you develop the most scientifically rigorous study, but acknowledging what your participants are able to do, what they're able to handle, and making sure that you're getting the best quality data that you can while respecting your participants and all of the ethics to consider. So, there's general research challenges. Obviously, analysis takes a long time and you want things to move faster. So there are those general roadblocks. But like I said, I think the hope that I can hopefully convey here is that we are making that progress. We have learned a lot of things in the last couple decades that we can really put to use now. And I think we have a lot of hypotheses to test and we have a lot of specific things to really dive deeper on. And I hope that that brings some hope that we are making some significant progress. And we have all these brilliant minds coming together to share their expertise and really, like I said, try to work together to solve this big problem.
So
And I think most important ways for supporters of OMF to help. I mean, gosh, we receive such wonderful support from you all, and we thank you so much for that. It really does mean the world to us. And I think, from the researcher perspective, I would say if you are able and willing to participate in research, that is so huge. We cannot conduct this without you. Your participation means everything to us. So thank you for those of you that can and are able to do that. We know that's not necessarily possible for all of you, but wherever you're able to support, we very much appreciate it. Linda, if you want to add anything to that too.
yeah, excellent. Well said. We appreciate the participations more than anything else because we couldn't do this without. But I need to add the other roadblock and the way we can support is obviously funding is always the roadblock. So we can only do what funding that we have. And so we have to scale our studies and take time and raise money before we can start large projects. And so funding is one of the biggest roadblocks that keeps us doing as many projects as we can do and holding us back to do some projects that we would like to do. But we appreciate all the support that we have had and we get from everybody all around the world, obviously, that helps us keep this going. So we just wanted to add that as well. So thank you.
And I think if you're not in a financial position to support, sharing awareness of OMF is
Yes.
also really important and helpful. Just spreading the word about what we're trying to do. And, it might reach somebody that does have the financial capacity that maybe you don't. So,
yes. Thank you, Danielle. When will the LDN trial end?
Yeah, the team is hard at work recruiting patients as we speak. I think we're about a third of the way through the total number of participants right now. So, it's a little bit of it depends on how many patients we can get through the door that match the inclusion exclusion criteria. But we're hoping by the end of 2026 to have all of the participants through the trial, and then we'll, of course, do the data analysis from there.
Can you summarize the recent study on sex hormones in ME?
Well, there are, I think, a number of studies on sex hormones. I think I'll maybe just broad sweeping, I'll say, in general, we are seeing some particular differences between people with ME/CFS and healthy controls for sex hormones, whether that is, looking at females or people of reproductive age and how those cycles change compared to people with healthy controls. Maybe there's some differences in those sex hormone levels. And then I think, generally looking at different pathway analyses, we're seeing some lower production of kynurenine, for example. That's, along the lines of one of these pathways that are connected to all the sex hormones. So, I mean, there's a lot of different studies in this area that I can't quite touch on all of them, but hopefully that gives you a good general answer.
And I think there's more studies happening now too, so we'll have some more information on that in the future, hope, very soon. Would be great to see more of these excellent sessions in the future, quarterly or half yearly. Thank you. Yes, we will be doing more of these because people like yourselves do like them, and we like to share what we're doing. And so we will be doing more of these. So stay tuned. Are there studies regarding MCAS and ME? We know MCAS and ME does not generally present in a dramatic anaphylactic fashion like other populations. It's difficult to get mast cell stabilizers prescribed for patients with ME in many places, and literature to support it would be good.
Thank you for that feedback. We don't have any projects specifically focused on MCAS and ME, but I know, it is a common comorbidity, and so that is generally collected information in a lot of our studies, and I think goes into a lot of the subtyping analysis that we're talking about. And there's a recent study that our Melbourne group put out that was looking at a lot of UK biobank data and trying to develop a diagnostic algorithm based on a lot of different factors and trying to differentiate it from comorbidities. So those kinds of studies will likely incorporate MCAS. We don't have any, but like I said, we don't have anything specifically focused on MCAS right now. But yeah, thank you for that feedback.
Do you think that antibodies like alpha and beta adrenergic and muscarinic, never know how to pronounce that word, mentioned by Carmen Schopenborgen, can be useful to find the type of ME that we have? I did tests and I'm positive.
Yeah, I think from a general perspective, things like that are really helpful for some of this personalized medicine sort of thing that I was talking about earlier. So, I think there are a lot of different types of treatments that people talk about in terms of ME/CFS, and they all have different mechanism of actions. So that's, how the drug actually works within the body. And so I think it's really important to have some sort of testing, like you're saying, this antibody testing, to kind of go hand in hand with some of the drugs that are trialed, especially when they have, a very specific way of working in the body. Some repurposed drugs can have some serious side effects. So, really working with your physician to figure out what might work for your specific situation is really important. And I think tests like that are a great example.
What will the subtypes classifications be based on? Blood, clinical picture, symptoms?
All of the above. Yes. We are hoping to use as much information as we can get. So, as Linda mentioned, these are previously collected samples, so we don't have to start from scratch with recruiting patients. But what was really important to us about using these samples was that we did have a really detailed clinical picture associated with each of them. So that includes symptom profiles, but it also includes a lot of other things, whether that be demographics or clinical labs or any other information that was collected. during the sample collection. So we have a lot of information to go through, which is partially why the analysis will take so much time. But yeah, really all of the above.
Will you use any biobanks?
Yes, as you know, we were just talking about with BioQuest, these are already collected samples, so we are using biobanks that exist right now. And so some of the samples are coming from Uppsala. They were collected and banked purposefully for research in the future. So we're using some samples there. And then we're also using some samples from the United States as well that were collected as part of the chronic fatigue initiative that the NIH funded. And so those are in a biobank right now, and we are working on collating both of those sample sets to do all the testing for BioQuest. So yes, biobanks are a fantastic resource.
And some of our researchers are looking at the UK biobank data, just looking at the data
Mhm.
itself and studying the data. Chris Armstrong in Australia has been doing a lot of studies on that as well. So, and our other researchers are looking at it. And then we'll correlate all those results with the results that we're getting from our studies. So biobanks are very helpful as far as the data and also as far as the samples that are available, especially for validating some of the studies that we're going to be completing. So good, very good question. Will you use decode ME data?
I think, in general, we won't necessarily use that specific data, but what we're hoping is that any genetic information that we are collecting or evaluating might be used to validate some of that. And, I think the Decode ME team is actively working on all of that data. So rather than, replicating work, we're hoping to complement and hopefully validate with some work that we have ongoing.
Thank you. Has there been any studies in periodostigmine in those with rapid emptying, with diarrhea and such?
Not that I know of specifically. I know our Harvard group has done some previous work on pyridostigmine, specifically looking at cardiovascular components of things. That's partially the area of expertise that they have. And so I think that was a lot of their focus, but they might have some clinical information related to this within that study. I just am not super familiar with that particular component of the clinical picture.
So these have been fabulous questions, and really, thank you so much. If there isn't any other questions, we'll probably sign off in just a few minutes unless you have any other additional questions, because we really love doing these, and we also love the questions. It helps us think, and it helps share the knowledge. And whatever questions you have, we know other people have similar questions, and it's good that you've asked them. We really appreciate you doing that. I think I see one more coming in. For those patients who are unable to work very actively but have scientific background that could be useful, are there ways in which we can contribute with with our particular knowledge?
That's a great question. I think we welcome feedback from everyone and anyone who is able and willing to give it to us. I think if you have a particular area of expertise and you want to be involved, feel free to reach out to us. We can see if there is, anything in that area that we have going on that could use, a person with lived experience. We want to incorporate the patient voice into everything that we do. So that is always very welcome, and we would love to talk further.
Great. Great. I think we're just at the top of the hour, and I want to thank you all. I see one more question just came in. Are there any other medications being trialed?
There are always medications being trialed. I think we have. I touched on this a little bit during the presentation, but we have a study up at Montreal that I haven't dived as much into in terms of the medications, but those are, looking at some repurposed drugs that are maybe less used in ME/CFS to date right now, but it's focused on some of the hypothesis-driven work. So those medications are definitely in the works. We're trying to do some validation studies to make sure that we have a good scientific evidence before we go through a really extensive and resource intensive clinical trial. But like I said, there's the personalized treatment trial at Melbourne that's doing a lot of different kinds of medications. We have the LDN and pyridostigmine at Harvard. And then a lot of the work that we're doing, is building up all of the evidence that we're hoping to be able to use for future studies.
And we hope to do some pilot studies. So we want to do some small studies to see if there's any proof of concept of drugs that are helping people before we do some really large, expensive clinical trials. So we are looking forward to doing a number of clinical trials in the next few years. And of course, it takes funding to do so. So we hope to, have enough funding to be able to do those kind of large studies. So we all hope to do more medications and trial more medications. So very good question. So I want to thank you all for your time and for your support and for following what we're doing. And if you're not already signed up to our newsletter, you may wish to do so just to keep on top of some of the things that we're coming out with. We like to keep you informed as much as possible with e-blasts and social media and as much as possible. And Danielle writes an excellent Science Wednesdays that we post on social media on Wednesdays. And so you may want to go on social media on Wednesdays and take a look at those as well. And if there's no other questions, I thank you all and wish you all a happy, healthy, calm, and warm holiday season. So thank you all so much. Take care. Bye-bye.
Thank you.
Thank you. Bye-bye.